Very good morning to all of you. Good morning to our respected speaker, Dr. Bide sir, all the offline participants and online participants. So I welcome you all on the for the second day of this training program. So our speaker is here, and today we are going to learn about the primer design. So our speaker is here, and I request all the participants, both online participants also, to use your laptop. And as per the suggestion of our speaker, so as he suggested that everyone should have the laptop. So all the online participants, they are also requested to follow the instructions, and whatever the task sir is giving to all of you, so you should go through it, and whatever sir is going to instruct us, so you should use your laptop. So thank you all. So over to our speaker, Dr. Bidi sir. And all the this trainees, if you have any suggestions or any confusion, you want to ask any question, you can write it in the chat box, and so that after the lecture, sir will take those questions and they will be answered by sir. So don't, and please keep your mic mute. Don't use your mic in between. If you have any query, you just put it in the chat box. Thank you.
Kapil. Yeah. Okay, let's start. Uh, Rajashree, can you? Her name is Rajashree, right? Anu, can you please ask them whether they uh, hear very well, because you need to unmute them.
Hello. Online participants, can you hear us? Can you hear me?
Sir, yes, sir.
And
Yes.
slide is visible?
Yes, it's visible.
Yes, sir.
Okay, thank you.
Okay, so let's start. Thank you very much. So, good morning again, everybody. So, again, as I yesterday said, there will be some basic principles of DNA, RNA, and because without that, you cannot understand the primer. And the PowerPoint presentation will show you only what kind of primer it will be. But the details of primers I will draw, okay? Because putting everything in PowerPoint presentation is absolutely makes no sense. It will make around 100 or 150 slides. So let's start. Okay, yes. Is working now. So I guess you know all the this guy is inventor of PCR, Nobel laureate. Yeah, so there was one question there. This mouse. Okay, thank you. So it's one of the most controversial figure in history of Nobel prizes. Actually, he refused first, but then all the colleagues said, no, you have to accept the Nobel Prize. This guy is why? Okay, this guy is actually very much fond of a free life, like you see in the photo. He is a biochemist, or he was a biochemist, and first he was a rocket scientist. He wanted to boost the rockets into sky. Then he also had a couple of paper in Science magazine, Astrophysics Philosopher, so theory of the astrophysics. And but then he studied the biochemistry, and he invented a PCR, which is really groundbreaking protocol or groundbreaking experimental approach to amplify the DNA. And he is very much a believer of LSD and ganja. He was used to have drugs, and with these drugs, he said that he was getting better ideas. So I'm not recommending you, but I have experienced effect of ganja. Actually, this is ganja is a normal word in Europe. It is not something what we think that ganja is something, but the marijuana is called as ganja or weed in Europe. My former PhD tutor was fond of marijuana, and he was amazingly good in the paper writing, once having and then writing paper. So, as I said, he is the most controversial Nobel laureate, and so let's see the PCR theory, what this guy, controversial guy, invented. So first, what he invented, a PCR, was it was not like a PCR machine, but he invented the chemistry. So the chemistry has your buffer, magnesium ions, then you will have water, of course, then DNTP. You know all what is DNTP, primer, forward, reverse primer, and polymerase. Okay, so these are the main components of any PCR. So you have a Taq polymerase, or you have proofreading polymerase, PFU polymerase. So if you want, I can give you a brief idea of difference between different polymerases. But what we use commonly is Taq and PFU polymerase. Can you shift to the whiteboard? Can I draw in this slide? Right this? Yeah. Just ask them. Yeah, I'm sharing the slide. So yeah, it's visible. So just ask if it is visible. If no, then I can change the. Okay, so do I need to explain the theory of PCR? Huh? It's visible, but do I need to explain you the theory of PCR? Really? Okay, so let's start. Okay. Right. So you have a double stranded DNA, and that DNA looks A, C, C, G, or G. Okay, and so on. Millions of base pairs. The human genome can have 1 trillion gigabytes of the nucleotides. In PCR, you have 3 steps. First one is denaturation. Okay, then you have annealing, and you have extension. So in case of denaturation, it's 2 strands, they separate from each other. So denaturation can be achieved by different ways. First thing is heat denaturation. So you just boil your DNA at 95 degrees Celsius, so 95 to 98 degrees Celsius, and it will denaturate automatically. Another way of denaturation is use of NaOH, sodium hydroxide. So those who are interested in making single-stranded DNA for their NGS libraries, they use NaOH instead of. Yeah. So let's come back to PCR. So in the first stage, these 2 strands are separated. They go apart from each other, and what you will have are separated strands. But these strands are complementary. Okay. So I will draw a little bit more here and a little bit more. Now in the second step, there is annealing of the primer. Annealing means binding of the primer. Okay. So you have 2 types of primer, or we call sense and antisense, right? Or forward and reverse primer. I don't know what you call the same, forward and reverse primer, sense or anti-sense primer. So the sense primer or the forward primer is complementary to the anti-sense primer. Okay, so it will sit here. And the reverse primer will sit over here. Okay, because they have the complementary nucleotides to this strand and to this strand. Then in the third step, you have elongation. So in case of elongation, the enzyme is required. Till now, you don't need any enzyme. Till now, there is only thermodynamics approach. That means thermodynamics is separation and annealing. You don't need any chemical for that. Only what you need is salt and temperature. That's it. Now you need to extend. So there are several enzymes in our body. So at least 3 or 4 are very important. You have DNA dependent polymerase, you have RNA dependent polymerase, you have RNA dependent RNA polymerase, you have RNA dependent DNA polymerase. So 4 different categories. Let's go back to DNA dependent DNA polymerase. That means when the polymerase sees that there is attachment of the 2 strands, please turn off your mobile. If there is I lost. If there is yeah, so please turn off mobiles, because then if the link is gone, then I'm done. So when there is a complementary attachment of primer to DNA, you have 2 options. First, this end, 5 end and 3 end. So both ends are free, right? Now DNA dependent DNA polymerase, when it sees that this end is free, the 3 end is free, what it will start? It will start to fill in the complementary nucleotides. So if it is here T, it will add here, perfect, C, G, G, C, tuck, 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 so on. Okay. Taq polymerase will stop here. The same way, in this side, again the filling will, or polymerization or filling or growing of the strand will occur, and from the one strand you will have 2 complete strands. Okay, DNA strand. In the next round, what will happen? Again, this will disintegrate into 2. This will disintegrate into 2. Again, the primers will sit. A Taq polymerase will do its work, do its job, and it will start to grow number of the copies of DNA. Okay, I told you in very simple way, but in practice it's quite complicated and quite complex. Okay, now below all these things, what you see here is this primer is very important. You have primer, good primer, the PCR will be amazing. You don't have primer, PCR will not be okay. And name itself explains primer. Primer means the one that starts, the one that initiates. Okay. So the primer, let's see what are the steps in the primer. Sorry, I want to go back to PPT. Quite complicated. Okay, thank you. So the DNA primer, in theory, the primer can be any single stranded DNA fragment. Okay, what we saw is you need a primer that can attach to DNA, or the DNA fragment that is attached to DNA, and that could be a primer. But the good primer is like good boyfriend. It should not be promiscuous, or it should not attach to anywhere else, but it should be extremely specific. It should be a specific attachment and bonding. So there are some typical features of the primer, and with this typical feature, the primer could be the best candidate for amplification. So first, a primer length and target sequence length. So the primer length means your primer should be between 21 to 27 base pairs. Okay, this is thumb of rule. I will tell you why later. And the target sequence length. The target sequence length means what you have here, the forward primer and reverse primer, what length it will amplify at the end. So the length of the PCR product. And that length of PCR product is also limited. You cannot amplify, for example, 30 kilobases or 10 kilobases with the help of Taq polymerase. But you can also use different polymerases to amplify very big fragments up to 30 or 35 kilobases, not base pairs, kilobases. Okay. With Taq polymerase, normal Taq polymerase, you can amplify around 5 kilobase, but in extreme conditions or scenarios, you can go up to 5 kilobases. The usual standard is 2 kilobases. That is 2000 base pairs is a limit, good limit for Taq polymerase. But Sigma and other in vitrogen, they also sell the high efficiency polymerases. Okay, so far is okay, primer length. And okay, now the primer length also depend on the GC content of the primer. So when you have 21 to 27 nucleotides, there should be optimum concentration of GC, G and C, guanidine and cytosine. Okay, why? Because what you see here, the bond between cytosine and guanine are 3 OH bond. Okay, OH bond. In case of thymine and adenine, you have 2 OH bond. So the energy required to break the bond in case of cytosine and guanine is higher, and in case of thymine and adenine is lower. That means you have in the melting theory, you have 4 into GC plus 2 into AT. That means here the energy required to break the bond between A and T is less, exactly half of the GC. So more GC you have, the more energy you need to disintegrate the primer. Okay. So you should not put all GCs, that will be detrimental. So the ratio of GC is 40 to 45% is the best ratio. That means if your primer is GCAT, TGCC or like that, so you should have good amount of GC in that. So as you know, now the GC content and the length of GC content governs the melting temperature. Okay, and that melting temperature governs the annealing temperature. Let's go ahead. So annealing temperature means, okay, annealing temperature means the temperature at which your primer attached to the DNA. Okay, that is annealing temperature. And anybody knows how much is annealing temperature if I have the melting temperature 60? Exactly. Plus or minus is, yeah, more or less 5 is there. There is a really complex formula behind this, but that formula gives you exact -5 temperature. That means if you have the melting temperature 60, the annealing temperature should be 55. Okay, so you start with 55 as thumb rule, but several times it doesn't work. Then you have to go for gradient. Okay, anybody knows what is gradient? Yeah. Okay, so far is okay. Okay, then runs and repeats. Runs and repeats within the primer and within the PCR products are extremely important. Okay, so runs and repeats. For that, I will need a new whiteboard. Okay. So runs means how many times you have ATT, 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 or GCC, GCC, GCC. Okay, how many times is run? For example, if the sequence is okay, so if the sequence is like this, so this is called as run. So for example, your primer is sitting over here. Okay, the sequence is G, C, A, T, C, and then it continues in the PCR product A. Then this is called as run, and that is a problematic because a normal Taq polymerase cannot feel A, and it will occur a slippage. It will slip, and sometimes it happens that because of that slippage phenomena, you won't have one A. So you may have only 4 or 5 A's, and that's it. And then it will go to another nucleotide downstream to A is like this. So in the amplification, what you will have not sequence 6 times A, but maybe 4 times A only. Okay, and that is really detrimental for the genotyping, and that is detrimental for the protein production, for example. So this is run and repeat. Repeat means A T A T A T, and so on. So you may have in the primer the same. Okay, so for example, your primer start over here and it ends with AAT, then it will not understand where I should sit. I should sit here or here or here or here. Okay, and then it will have promiscuous primer, and the primer product or the product of PCR will not be unique. You will not be of the same size, but there will be a different products, or there will not be product. So this is called as run and repeat. Okay, so what you have to do is you have to check while you are constructing the primer, you have to check that particular DNA fragment, and then you have to see whether there are some runs and whether there are some repeats, and don't take into consideration that particular fragment of the gene. Okay. Okay. The cross homology is extremely important feature for the primer design. The cross homology means whether the primer is going to sit to other species, other genus, or within the genome. So for example, you have, in case of eukaryotes, those who are working with plants, or those who are working with parasites, you have alleles, right? And you can have different alleles, different repeats of the allele, or different repeats of that gene. So you have, it is called as repetitive genes. Have you heard about repetitive genes? No. So in leishmania, for example, or in different parasites, you have repetitive genes that we don't have in bacteria or viruses. But in those who are working with parasites should consider this small. So in this case, what will happen is you have repetitive gene. So the sequence is repeated 1, 2, 3, 4 cassettes. Okay, and then what will happen? Your primer will sit here, 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 and here as well. And just because of the repetitiveness, you will have PCR product like ladder. Okay, so the cross homology is within the genome. This is a repetitive gene. And orthologous cross homology means between the, for example, if you are identifying any bacteria or parasite from the host, okay, then you may have a cross homology between the parasite genome and the human genome or the cattle genome. So you have to avoid that species cross homology. Okay, for example, which genes could be a homologous? Can you name one or two? For example, if I am working on identification of parasite, I should not, from the animal, I should not make primer on 20S RNA or 18S RNA, because those rRNA sequence is the same in parasite and in mammals. So you should not take into consideration. Although this is very good gene for the identification, no. Secondary structures, extremely important feature. So just write 3 points and then tell me one by one. So you have hairpin, you have self dimer, and you have cross dimer. Anybody knows about this hairpin cross dimer? Yes. Yeah. Rashmi is saying yes. Yeah. Okay. So I will draw and you will tell me what structure it may make. Okay. Okay. So you have, for example, this primer. This is A, C, T, G, 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 A, T, A, G, T. What structure it will make? So that is called as, yeah, this is nicely complementary 3. So that will make this kind of structure. Okay, so that will make okay, and that is called as hairpin structure. That what you use, girls, you use hairpin, so same hairpin structure. So what will happen is the hairpin structure, if you have 3 nucleotides, the melting temperature is less. If you have 4 nucleotides, the melting temperature is high. It also depends which nucleotides are in the structure. For example, if you have CG, CG, then you are done and hairpin is very strong. What will happen is hairpin will remain in the PCR reaction Then there is separation of the fragments in the denaturation. In the annealing stage, when the temperature is going down, this structure will remain like this, and there is no free nucleotide to attach to your DNA, and PCR is negative. Okay, so this is called as hairpin structure. Okay, now the second one is self-complementary. So, for example, you have we are calling self-complementary, right? You have G, then you have A, T, G, for example. Okay. So what may happen that one primer is like this, the forward primer, and the same forward primer can have one more. I have to add GC. Okay. GG. Yes. CC. And here we will have A and T. So this is a typical feature of self attachment or self complementary. So in this case, what you have, CCC will go and attach somewhere here or somewhere here. So it will not be a perfect hairpin like structure, but like this. Okay. So self complementary structure, and at the end it will look like this. Oh, so it depends at what stage you have. So for example, if you have GG here instead of AA, it will go like here. Okay, so this is called as self. Now the third one is cross. So you have two primers. First one is forward and second one is reverse. So for example, you have, that is but bit simpler. You have AAA, TTCGG is a forward, and if you have in the reverse CCGA, then they will attach to each other and they will not separate as such, and they tend to bind very faster than to bind with the DNA. Why? The forward and reverse primer tend to attach to each other faster than they attach to DNA. Can anybody tell me? No. That is true. The length of nucleotide is that is true, but amount of DNA or copies present in the reaction is higher. Amount of DNA present in the is lower. So for example, if you put 100 nanograms of the DNA in reaction, okay, 100 nanograms, the complementary region is very less. The DNA is high, is big. Complementary is less. So primer has to find where is that, and it has to attach. But if primer sees that, oh, my colleague is here, that is complementary here, it will attach to that colleague. Why he will search for the complementary? Yeah, so simple theory. So then this is a cross attachment will be faster. So you have to avoid this cross attachment very well, and that cross attachment could be 2 nucleotides, but not more than that. Okay, so all 3 points are clear for you. Now, these are not included into the how we should design the primer, but these are my experience. You should not have A at the 3 end of your primer, and you should have G and C at the phi end of your primer. Okay, so that means if the primer is like the forward primer is like this, you should not have here A. You can put T, C, G. Best is C, best is a G, because it will have very good binding to complementary. Okay, but this is my experience. And very important is when you purchase the primer, it comes lyophilized, I guess, right? And you put the water to make the 100 micromole or picomole solution, and you make again dilution 1:10 or 1:3. I make 1:3. Usually I don't make 10 picomole, but I make 33.3 picomole solution, working solution. And that solution I aliquot. I do not freeze thaw, because then there is a breakage of the fragment. So once you have the lyophilized powder, put the required amount of the water, and then make the working solution, and that working solution you can freeze thaw. That working solution you can aliquot, and then you can freeze thaw. Okay, I tell my students to have limit of 5 or 6 cycles of freeze thaw. That means one aliquot you can freeze and thaw 5 or 6 times, not more than that, and you throw it away. Sorry? Water.
Water. NFW.
What you say for that?
Nucleus free water.
Nucleus free water. Okay. Yeah, water is important because when we produce the primer, I don't know, you know, the primer synthesis chemistry, but once we produce the primer, we desalt it, or we make HPLC, or we make SDS phase purification. So there are three purifications. The machine will give you primer. You desalt it with the column, okay, or you purify through the HPLC. And then once you purify, you have to put in some buffer, right? And we always use TE buffer for that. And then we lyophilize it. But TE is not a volatile buffer, so it remains there. So your job is to put water, and then again it will reconstitute into TE buffer. So you don't add more TE, otherwise what will happen? You will add more ions in. Yeah, but it is automatically in that. Yeah. And if you buy from Sigma or other companies, Biotech, for example, so they add T buffer, and they recommend you to add water, not the buffer. Okay. So far you understand the simple primers. Now we have two options. It will be more difficult. Now I would like to have feedback from you. For example, which software you use, or you don't use any software for primer design, right? A primer silicon. Okay. Now I would like to ask you some theory. For example, I would like to amplify or identify different pathogens from mastitis. Okay. So I will have 6 or 7 common pathogens from mastitis, like staphylococcus or streptococcus, and I don't know other. I forgot this mastitis, but you will have 3, 4. So what you do in that case? Do you use different primers? You use multiplex. Okay. For example, those who are working in viruses, flavivirus, for example, you have Casnor-Forest disease, you have West Nile virus here, I have tick-borne encephalitis virus coming from Russia over here, Japanese encephalitis is in India. All those are flaviviruses. Yellow fever. Okay, so you can have a diagnosis of infection, but in that geographic region, you can have 3 or 4 similar kind of viruses. The same thing goes with herpes virus, for example. And in that case, what you can do is you can make uh, generic primers, not specific primers, but it's called a generic primer. Okay, so it will attach to uh, DNA from different viruses. I'm not talking about multiplex, only single primer. Okay, so it will attach, for example, the same primer will attach to yellow fever, to the West Nile, tick-borne, and so on. Idea is good because you don't need to have multiplexing. Because multiplexing is really difficult, and there is a lot of cross contamination or cross amplification. Okay, so sometimes it's better to have a generic primer rather than multiplexing. Okay, thumb up rule says me that when you are going to have uh, this generic primer in the genome, you should find some homologous uh, ends or some homologous. How it looks, I will show you. So the idea behind is that I want to characterize, identify, sequence, and genotype that particular pathogen or infection. So what should I have to be able to amplify? I should have my primers in homologous, and within that there should be heterologous sequence. Okay, explain you. So this is okay. So this is genome. For example, we can take flavivirus again, and in case of flavivirus, you have a protein E, and that protein E is polypeptide or a whole genome. So protein E is highly heterologous, and before protein E is, I think, M protein, and that yeah, so M protein, and that M protein is homologous within different flaviviruses. Okay, and this region is heterologous. So the idea is you have to have the primer forward here and reverse here. Okay, so that will amplify different viruses, West Nile, tick-borne, and so on. And then when you will sequence, you will have exact identification of the particular virus. Is okay? Sometimes it is not possible because you will have some heterogeneity over here, and that heterogeneity is not big, but it's smaller. For example, you have 5 to 10% heterogeneity. That means you have, if there are 20 amino acid 20 nucleotides over here, you have 10% heterogeneity. That is, sometime in this will be A, here will be T, here will be C, again A, sorry, T, T, T. So if you make the phylogenetic or the consensus alignment, you will see that there is some heterogeneity, some small heterogeneity. So how my primer should be in that case? My primer should be degenerated.
All the participants are requested to mute yourself.
So my primer should be degenerated primer. Anybody can guess what that could be? Degenerated primer. So degenerated means when you have you are ordering the primer. Okay, you order for example, you send to the company, I would like to have A, T, C, C, G, E, or like that. But if you see here A, T, C, T, A, so most of the nucleotides are present here. What I will do is instead of T, I will order N. Okay, in case of T, A and T only, I will order Y. C and G, I will order M. Okay, so the primer that will come to me will contain A, T, C, A, C, C, A, G, C, A, T, T, all 4 combinations. So this primer can attach to any of the virus. So that is called as degenerated primers. So this is a very good alternative to the multiplex. You can go for some homologous part, but if there is heterogeneity, yeah, please. One more time. What you understand so far is homologous and heterologous part, you understand? Yeah, but within that, do you understand the homologous and heterologous part? Again, okay. So I will make it short, but again. So in a normal PCRing, what we are doing is we are going for the multiplex PCRing, right? Primer separated for this, separate primer for this, separate and so on. But I don't want to have multiplex. I want to have one single PCR for several pathogens at one shot. So my option is to find, first align the genomes of each pathogen. I have to check where is homologous part and heterologous part. And within that homologous part, I will see that how many percent of homology is there. You will never have 100% homology. It's very hard. So you will have, if you will have 90% homology and 5 to 10% difference heterology, that is your region. And in that region, you have to make the primer. I will explain you everything. Okay, hands on. Now, when you align this, you will find some heterogeneity within the sequence. And for example, if the heterogeneity at particular locus or particular site is A, T, C, T, 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 okay, at that particular, you don't need to order a different primer, but you need to order primer like this. If there are 4 different nucleotides at same point, you order N. Company will give you mix of all 4. If there are only 2, it depends on which 2. You can add Y, M, and I will give you this table later. IUPAC table it is called as, and that table tells you if there are A and T, what letter you have to put. If there is C and G, what letter you have to put. If there is G and T, what letter you have to put. So all, if there are 3 combinations, C, G, T, what letter you have to put. So all the combinations are there in the table. You should have table in front of you and order the primer. Got the idea about that? And I'm using a lot these degenerated primers to save the time of multiplexing. Sorry? Chemically. N is actually, you get the tube, right? A company has to produce 4 different primers. They have to mix together. Company means that the instrument makes 4 different, and then it adds equimolar of each primer in one tube. I'm talking about only one degeneracy, but it's extremely complex. So for example, you have here A, N, P, W, C, G, M, P, C. You can see the combinatorial effect behind that, and you will end, for example, you have 4 into 3 different combinations into 2. So that many different primers you should have in single tube, or even more primer. Yeah, it is combinatorial effect of producing the primer. I usually order up to 4 degeneracy. Okay. Yes. It will affect the annealing temperature. So the degeneracy is depends for what. We use degeneracy for production of antibody, but we also use a degeneracy for generic PCR. In generic PCR, what you have to do is annealing temperature, for example, is 55, because melting temperature also affects. So if the melting temperature, highest melting temperature is showing 60, the predictive annealing temperature is 55. You put annealing temperature 50. Again, 5. So you go to the lower level. So what will happen is there is more chance of attachment. You will see everything this. So write down this point, degenerating primer. I will show you in the software. What you have to also tell me is homologous and heterologous regions, alignment, classical primer design, and based on the information what you got, you have to tell me the explanation of each and every component when I'm going to design the primer. Okay. Okay, so I already explained you the next point, which I wanted to explain you is target specific versus generic. So you got the information about target specific, the exact one target, generic within the different genera. Now this is extremely important aspect, which gene I should take into consideration for preparation of primer. Okay, either I should use the housekeeping genes, right? These are some of the housekeeping genes, or antigen specific genes. So if I want to use or if I want to make target specific or species specific primer, I want to use a species specific identification PCR diagnostic kit. Which gene I should take, this or this? In case of bacteria, and this is in case of parasites, I kept both. Okay, or so in case of bacteria, which gene should I use? 16S rRNA for species specific identification? No, sorry, yeah, exactly. And I already explained you, when there is infection of parasite, infestation of parasite, I should not use these two, because host and pathogen both will have the same. Next point is we already covered this point, that is a product length. Okay, so long or small target, what should be my cutoff? So for example, if I want to send the sample for sequencing, what should be my cutoff? Anyway, what should be my product length? How much? For no, I'm talking about Sanger sequencing, not NGS. Yeah, around 800. So Sanger sequencing by default, the cutoff is 1000 base pair. It depends on what capillary you use, but what we use is 50 centimeter capillary in the Sanger sequencing machine, and by default it gives me 600 base pair maximum. There are 80 centimeters capillaries which can give me up to 1000 base pair, but it is not usual. But several companies in the world, they use 50 centimeter capillaries, and that by default gives me only 600 base pair sequence. So it is useless to make bigger product if you are not going to sequence it, right? Okay, so for sequencing, anybody knows what is HRM, high resolution melting analysis? How many of you have real-time PCR in your laboratories? You have. So you know what is HRM. Sandeep, you should have. You are using HRM. So what is the product length maximum in that case? Sorry? Product length, short. So should be 150 base pair. Okay. So this is the maximum product length what you can have. 200 is fine, but you will not have distribution between different products. So it depends what is your downstream analysis. So the answer to long versus small is, what is your downstream analysis? Are you going to do single strand conformation polymorphism? Are you going to do DGGE? Are you going to do HRM, high resolution melting? Are you going to send it for sequencing? Are you going to do fragment restriction fragment length analysis, RFLP, and so on? So for RFLP, there is again different design, not only product length, but how many restriction enzymes that product contains. Okay, so that is also, so answer to this point is, what is my downstream analysis? Okay, so we will stop here, theory, and we'll go to the first hands-on, but from my side, you will see, and then we will have a break. You install, and then I will give you assignment. Okay, and then you prepare a primer of your own choice. I would recommend you to prepare primer which you will need. Okay, you don't make any kind of primer. And again, I forgot to tell you why I'm recommending, or why we are constructing our own primers. Yesterday I told you a little bit, but new genospecies are coming, or whole new virus is coming, for example, COVID. And the existing primers which are already published, maybe you will take from 2000, 2004, 2005. But these new genus or new species are not included by that primer. But whenever some new species come up, they have to send the information to gene bank, right? And that data you can retrieve while constructing the primer. So for example, if there is some new species in India, you will catch that species because you constructed your own primer, and that will avoid a false negative. If you take some primer which was constructed in Latin America, where you don't have a species in India, homologous, it will not catch, it will not amplify a species from Indian subcontinent. And then you will have, oh, I am free of paratuberculosis, but you are not. Okay. So that's the main problem what you have. So you have to go for generic primers first. You have to make your own generic primers. And then if you want to make save time, for example, then you can order which are already published. Okay. So this is one point, not to miss any or not to have false negative result. Then because of the generic primers, you may discover some new species because of the generic primer or because of the degenerative primers. You will discover some new species, and that new species will be yours on your name. So whatever primers we are producing, we produce at least 600 to 700 primers per year for our laboratory, and all the primers are evidence based. And we also have use for genotyping and so on. Okay. What I recommend you, if you really want to use primers, don't use from the publications. It is not good idea at all. For that, you can collaborate with me or our guys, and they can design primer for you, no problem. It is, and primer design is like drugs. Once you catch that, you will design always, and you will never go back to the used primers. Okay. Okay, so just 2 minutes give me to start the Genius. Any questions so far? Yeah. Yeah, sure. Yeah, yeah. Look, this is a good question. How to avoid this non-specific amplification? With the help of bioinformatics, first you have to check whether this primer has some complementarity to the genome of cattle, of human, of the host. Okay, so if you make the bacterial the primer based on the bacterial genome, always you have to check the cross reactivity. After doing that, there is only 50% chance that it will be specific. If you have unspecific primer and you get multiple bands, do not play, throw it away, order new. Order new means order new sequence, not the same sequence. Order new sequence. What? No, no, no. You don't need now your laptop. First, you just see. Girls, come over here if you don't see it from there. Okay, so instead of showing you the already designed primers, you can come over here. Sandeep, you have there. Very good. Okay. You can tell me which primer I should start with. For example, give me some gene or pathogen, better pathogen, right? Because you are working on the pathogens, right? But give me bacteria better. Sometimes parasites are very complex. Coxiella. So let's go for Coxiella. So I will first show you Genius. So what you see here in this part, this is your stored files or offline files, and then this here you will work in this, and this you have some guideline. I never read this, but it is always there. Then you have here different databases. So what you see here, this is NCBI database. Everybody knows NCBI database? Yeah. No problem. So we actually started hands-on now. So NCBI database, how many of you have submitted your gene or your fragments in NCBI database? 1, 2, 3. Yeah. So you have idea what is NCBI database. And it is not possible to publish without submission. You know that. Okay. So that data we retrieve. So in NCBI database, there are tens of different sub databases. First is gene database, you see here, everybody. Then gene means all your gene fragments, what you identify with PCR. Genome database, so all genomes are there, right from human. Nucleotide database, this is also nucleotide, but maybe short, long, non-curated, and so on. The gene database, I will go back, contains a gene open reading frame or protein coding sequences, not intergenic spaces or the non-coding sequence you will not have in gene. Then this is I will just keep PubSet. Then it is protein database, PubMed. You know all PubMed, SNP, single nucleotide polymorphism databases, and the structure of the gene and proteins and the taxonomy is there. So today what we'll need for Coxiella burnetii is either genome or gene. Okay. If I know the genome, how long it is of Coxiella. Coxiella is a bacteria, right? So I should click to genome, and one more database I forgot is UniProt, that is protein database. So I have two options in this case. If I know the exact gene name, I can retrieve the gene. If I want to amplify the gene, I should go to the gene. If I don't know what I should amplify, I should retrieve the genome. Let's go to the gene first, because genome is, it will take a while. And okay, so I have here, Sandeep, can you please tell me gene? COM1, COM, and enter. I hope it will work. Yeah, it is searching. What is that COM1? Sandeep. COM1 is the full form of COM1. Okay, so we have one DSBP protein, for example, and what we need is Coxiella burnetii COM1. Okay, I forgot to add the Coxiella here. Okay, so now you see there is COM1 and Coxiella. Enter, and it may give me where is Coxiella is here. Yeah, should I take a OMP, outer membrane protein? Yeah, there is. So what I should do is I should click on download. Okay, so far you understand what I did. I click on the gene here, then I click here, I put some name, but Coxiella burnetii, and then I have here Coxiella burnetii and the outer membrane protein, for example, and I will put download. And where it is.
आपने शुरुआत की 2-3 लाइनें क्या बताई थी आपने?
You can ask question. Sorry. No. Should we start? Yeah. I lost the Coxiella, so I'm searching once again. Okay. I'm just downloading the sequence of that particular gene.
So, sir, one minute, sir. Sir, online participants, I hope you see the screen. Screen is visible to you?
Yes, it's visible.
Yes, sir. It's visible.
Okay, thank you.
Okay, so after searching the outer membrane protein of Coxiella burnetii, what I did is I downloaded the sequence. Perfect. I downloaded the sequence, and this is the sequence, what you see here in yellow. And that sequence, what I will do is I will just hold that and put in my folder, for example, in Mumbai. Okay. So you have the sequence over there. That sequence tells me it is the sequence length is 498 base pair. Okay, topology is linear. Accession number, you have how many GC content it has. Genetic code is bacterial. Genetic code means the coding, the translation from RNA to protein. So there are different codes. So coding is bacterial. The genetic source is chromosome. So it is not coded by plasmid in case of bacteria, but it is coded by the chromosome. So all the information is also automatically retrieved. What you will see here, this is a sequence. Okay, what this? The second folder is annotation. The text view, whatever you see from the gene bank, similar kind of text view you will have here. Then the FASTA file. The FASTA file is just sequence. Do you know how to make the sequence into FASTA? You just put this sign and that's it. And the whole information is over here. Again, name, which bacteria, Coxiella burnetii, which strain it is, RSA 493, and the whole taxonomy is also included over here. So this is a rough picture of what you downloaded. Okay, so I will go back to the sequence view, and if I click here, I will start to see the sequence. Okay, it's simple. Now I would like to have, I would like to make a primer. I would like to use this for the primer design. The simplest form of the primer design is primer. So click on the primer here, design new primer, and this window will pop up. So based on the theory, what you learn, we can go one by one. Okay. So first, which region I should include? That means I should tell this software how big should be my product size, or which region I should include. So you can guide me. For example, there are three options. First option is include region from 1 to 500 or to 490. There is another option, target region. So I can give from nucleotide number 20. Sorry, my in Slovak. So 20 to 200, for example. Or there is third option, is a straightforward option, is product size. So do you remember I explain you the product size depend on your downstream analysis, whether you want to go for sequencing or HRM. So if I'm going for HRM, high resolution melting analysis, or sequencing as well, my product size should be within 150 and 200. Okay. No, target region I didn't click, so it will not taken into the consideration. Okay, number of pairs which should be generated. So this software can generate 10 pairs, 10 different pairs, and so on. So let's go with 3 different pairs. You will have idea. If I will put 10, it will be a mess. Then TM calculator. So either Santa Lucia or Bersul. I never use this. I use always Santa Lucia. Which concentration I'm going to use, monovalent or divalent? I usually use monovalent buffer. Okay, so 50 millimole is perfect. The oligo concentration I use 50 nanomole, and DNTP concentration 0.6 millimole I use. So these are the values which I have stored there. So that's why they are appearing. I should remove those. So you will tell me. Okay. The size minimum and maximum. I told you 21 to 27 or 30. So 21 to 30. So optimum size is 21. I can put 25, for example. So in between. Okay. So this gives me idea of size of primer. Okay. The size of primer. What will happen if I have 10 base pair primer? Why I need 27 or why I need 21? What will happen? Sorry? Why mispriming? Is perfect answer, but why? Yeah. So more is the length, more is a melting temperature, more is annealing temperature at one side. Another side, more is a length, more specific is a sequence. So more specific is sequence, more chances of mispriming. Okay. So 21 is optimum or 25 is optimum. Again, TM, what TM should I have? Any idea? What will happen if I will have TM50? TM50. TM means, okay. What if I have TM50? Annealing temperature will be 45, and what will happen? Binding will be there, no problem. If it is 30, binding will be there. Yeah, less is your temperature. I showed you the self dimer, and hairpin, right? So in case of hairpin, you have complementarity of 4 or 5. It also has some temperature, right? And that complementarity has a lower temperature because there are only 4 or 5 nucleotides, right? So to make the hairpin straight, you need more temperature, and that temperature should not match with your annealing temperature of the primer. So that's why your annealing temperature should be higher, and that higher is about 58 to 60. Okay, it's extremely important when you are doing real-time PCR. Remember, I may forget later, but in case of real-time PCR, you can use only two-step PCR. First, denaturation, and combine annealing and extension at 72. In that,
Sir, voice is not coming.
now, so the melting temperature of forward and melting temperature of the reverse should be similar. Why? because if there is big difference at the annealing temperature, one will anneal, one will remain happy in the solution. Okay. So they should have similar melting temperature. In other words, there should be the TM difference should be as low as possible. The maximum dimer TM, the next parameter, maximum dimer TM is 10 degrees Celsius. Okay, that means if I put here 60, it will allow to have a big dimers. If I will put 10, it will allow only 2 or 3 complementary regions. Okay, so I will put 10. Maximum dimer TM, sorry, 3N stability. Now 3N stability, what I showed you or told you in my experience, I never put A adenine at 3 end. Okay, so if the primer, forward primer is like this is 5 end, this is 3 end, right? It will grow like this. In case of reverse, this is 5 end, this is 3 end. It will grow like this. Now, the maximum 3 end stability, if you put adenine, there is less stability. If you keep G and C, is higher stability. Okay. So in numerical values, it tells me you keep 13 to 20 the stability. You just write down. The explanation will be quite longer. Okay, now my job is to say okay. Sorry. Yeah. So that is fine. TM is fine. It should be 10. So now you can see there are different primer pairs. Okay. So this primer belongs to this. This primer belongs to this. If I go here, come on. If I go here, here it will be highlighted, right? So the pair is this one. If I make some bigger picture of that, okay, so it's bigger. So this is my forward primer. You can see here its properties. Name, you can change the name later. The length is 25. The interval means from which nucleotide to which nucleotide in that particular gene. GC content is 44, TM is 60, hairpin, no hairpins. Very good. Self dimer, no self dimers. Primer dimer, no primer dimer, and sequence is this one. And the product size will be 158. Awesome. Okay. Yeah. Just a minute. I will go back. Yeah, here. Should I put one? Yeah, no problem. So you will give more stringency to that. It's the same. So it falls within that stringency. In case of reverse primer, you have these values or these properties. So TM is 60. Let's see what is TM of the forward. 60.1. So difference of 0.1. Okay. GC content only 36. not 40. What we said that should be at least 40, but that reflected into length. We don't have 21, but we have 25. So is longer. That's why the TM is 60, but the GC content is lower. That is automatically adjusted, no problem. Again, hairpin, self dimer, pair dimer are none, and the product size is the same. I mean, should be here somewhere, 158 product size. So this is one pair. Another pair is this one, and his brother is here. Okay. Let's go to some other primers. For example, new primer. And now what I will do is I will go for 300 to 490 maximum length, and let's see. So wait, I will remove all the primers. Okay, again, primer, new primers, okay, and 300 to here, 300 to 498, because the size is 498, and click OK, and you have you have these two primers. First one is this primer. Again, see the characteristics: 60.1, none, and product length is 325. Okay, here is the same. Okay, so it's brother 60.1, so difference of 0.1, 325. So far is okay. So in this case, you predicted, or you made the primers. Now how to order the primers? Okay, what I will do is, I don't know how you order the primers, whether you
All the participants are requested to please mute yourself. It will disturb the other participant.
Okay, so thank you. So I don't know how you order the primers. From the publication, you see the sequence and you order, and I guess you put into the Excel sheet, or you send it to company by email. And for example, if I want to order this primer, you see the sequence here: CCG, CCG, CAG, and so on. Upper, okay, CCG, CAG. Okay, this is forward. But don't make mistake while ordering the reverse primer. That is extremely important. Let's go to that. See here. If you order your primer like TCT, CGT, you will never get the PCR product. You have to reverse complement the sequence and then order. So the software make it for you automatically. TAC, so reverse complement to this TAC GGT, TAC GGT. So you go here, copy the sequence, go to the Word file, paste it there, order the primer. Yeah, it's simple now. But if you copy this, then you are done. Okay, so this is a simple approach to have primer. Now, yes, 300 is very nice. 300, yes, it's fine. Even 350 is amazing for sequencing. It depends how heterologous is your gene. More is homologous, more length you have to cover. More heterologous, less length you have to cover. More heterologous, less length you have to cover. Okay. More homogeneous gene within the species, bigger picture you need. Okay. Okay, now to your question, what you ask, if I have primer, and what about it non-specificity, how I will detect that. So for that, for example, or whether that primer is okay or not, this is the first question. And the second question is whether it is non-specific or not. So for example, I have the primer from the book. So I will just remove. So I have a primer from the publication that tells me that this primer is used to amplify the outer membrane protein H of the Coxiella burnetii. And I just want to check it. I want to check its characteristics and so on. Okay, whether it is good for me or not. I want to test both. First, start with the forward primer. So the steps are the same. Either I should go with the blast online, or I can go with this as well. Blast will give me nothing. This will give me some more information. So I will go to again primer design, and we saw the first one. We saw design new primer. So we design new primers. Now we want to test the primer from the publication. So this is test with saved primer or test with published primer. I will click whether this primer is forward or reverse. So I will keep forward, and sorry, before that I have to first make the primer. So for that I have to go to the sequence. Sequence, new sequence, and the primer which I copied from the publication is this. So what I did is I went to the sequence. Here is sequence. Yeah, I click here, I click new sequence, and this window popped up. Okay, what I will tell from the publication, forward. This is a nucleotide sequence, or this is a primer sequence actually. So it can give you option, either you can put nucleotide or primer. I will choose the primer and I would say yes. Okay. Now this is a primer which I have now created. I will go here, test with saved primer. And where is Mumbai? Yeah. So in Mumbai, I have from the publication. Okay. You understand? And I will say select. And just I will put OK. This I will remove. I'll put OK. And this primer is now showing me it will attach somewhere here. Okay. And it will tell me automatically now the characteristics of that primer which I took from the publication. So TM is only 58, GC content only 27, but the length is quite big, 29. Okay, so theoretically, the length is bigger, not 27. It will, you have to pay more first. The second one is less GC content. Although there is no hairpin, self dimer is good. So 95% this primer is good. It has homology with OMPH, but only drawback is the less GC content. Okay, so in this way you can test whether the primer which you are going to order is okay or not. So simple test it is. The same with reverse primer. Okay, now when I do the protein production or recombinant production, I cannot use this kind of tools because I need specific sequence. Region it is good or which region I want. So for example, let's go for that. I want, for example, the sequence of the protein, this one. Okay, so sequence of protein start M, methionine start codon. I should have primer over here. And what I taught you that the primer length should be 21 at least. So let's see here. What I selected has 21 base pair. You can see here down. Yeah, here you see 21. So it's 21 base pair. Now, more or less, you can see in this region, you can see the TM is only 50. So what I need to do, I need to add more nucleotides. The TM is 54. TM is 54. It's still less. TM is 58. I'm happy. Okay. So this I would like to have a primer. What I will do is I will just tell him convert to oligo, primer, convert to oligo, primer. The sequence being converted into oligo could not be characterized. Okay. Okay, let's go away around. So methionine again. This sequence I would like to have as a primer. I will extract this into new sequence. So this is a sequence I want to convert into the primer, and I will tell convert into oligo and okay, and that's it. It converted into oligo and automatically tells me if I go here the characteristics of TM is 72, cell dimer 18, hairpin none. But I'm not going to use this for the classical diagnostic purpose. I need to tweak the PCR reaction to get rid of cell dimer, but this primer will work to amplify, but not what you can do is you can close that door, and this door is not giving any sound, so this keep this door open and you just don't use that door. Okay, so what we saw is we can either tell software to make the primer, we can ask software to analyze our primers which we are taking from the publication, or we can make the region of interest and convert into the primer. So 3 different options we have. Now, if you want, you can download your sequence from the Genius. This is really basic training. It is not advanced training, but it is better if you work with that. At least I will have 5-minute break, and you will also learn how to make the primer. Don't use any make the primer of your interest. Okay. So 10 minutes are enough for you? Or? Okay. You don't need key now. It is free. Yeah, trial. You put trial. Yes, request. Huh? Yes. Yeah, trial, but I can give you to activate later. No problem.
So online participants, we will have a 10 minutes break and then we will continue.
बघितलं मला सांगितलं म्हणजे इथं म्हणून आहे ना गोष्टी मला वाटतं इथं असते. कोण म्हणत होता विचारतो हा बरेच जुने विचारतो कुठलं ते काय म्हणून बोला विचारतो मी
बिल्कुल ना okay okay so you can share the laptop those who are successful you can share with your colleagues
चेयर लेके इधर कर दीजिए और यहाँ पे लगा दीजिए। दीदी
शुरू करेंगे। हो गया संदीप राव डन। Okay, let's start. At least you can share. So if in that group you have one computer, is okay. You were successful, I guess. Here, one is successful. So you can make some groups, and you were also successful. And here Rashmi was successful. So maybe 2-3 people on one computer will be easier then. Okay. So anybody is without computer? Okay, now let's go to the next level. Is like a video game, next level. Okay, fine. Because you put product TM 60, it is never going to happen. You know, I mean, whole TM, it's a lot. Karaga ka shuru? Okay. All ready? Ready, Kanai Azun? Yes. Here you go. Okay. So online participants, should we start? No response. Can you ask online participants? Okay, so at least we struggle with the software, but believe me, the struggle is will be fruitful for you in the future, because this software will allow you to not only make the primers, but search, download genomes, download proteomes. If you are going to do NGS, the whole NGS analysis. So it is all in one software. So if your university is going to purchase this, it will be most useful for you all and students. This software can be purchased as a one software for whole university, not individual. Individual costs 2,500 euro per year. So for whole university would be great from project, for example. Okay, so try to push that idea. Yeah, dollar. Okay, so let's go to next level. That means we downloaded the specific gene. We tried to make a simple primer. Okay, and next level is let's see within the species or within the genera, we can make some generic primer. Okay, so those who have activated, they can also later. but they can download. For example, let's talk about flaviviruses. Yeah, because that is most simple and smallest genome is only 11,000 base pair, and that will take less time. If you go to herpes virus, it's big. If you go to that pig virus, it is small bacteria. African swine fever, yeah, is very big genome, and it will take half an hour to download. So just to save time, you can later download the flaviviruses. You can write down genome of West Nile virus, West Nile tick-borne encephalitis virus, TBEV, yellow fever, Casanur forest disease, West Nile I said, right? Yellow fever I said. Any virologists over here? Japanese encephalitis virus. Japanese encephalitis virus. So that will give you an idea how to compare genomes. Don't try to compare genome of bacteria. It will be a lot. When you will have a little bit experience, then you can go for this hard work. I have already downloaded that because we were working on that. So I have that data over here. And then we can make the homology. And the homology, principle of homology will be the same what I'm going to teach you now. Okay. So, or alignment. How many of you know alignment? How to do alignment? 1, 2, 3, 4. Chaudhary Sahib. Alignment, you know. You know how to do the alignment? No. And you know? Okay. So why we do alignment? You know? Why we do alignment? And that reflects, yeah, and that reflects to the phylogenetic analysis, right? So how far is that particular strain from Latin America, from Europe, from Switzerland, from Russia, and that gives us information that how this infection might have come and from where, from farm to farm, from veterinarian to the farm, or from birds to the farm. Okay, that can be tracked based on the phylogeny, and that phylogeny is dependent on homology, right? Now, how many of you have done the phylogeny? 1, 2, 3. Okay, so phylogeny, let's see first the phylogeny. That will be easier. So in case of phylogeny, what I will do is you don't see. So those who are in the server, they can put in the search protein E flavivirus, protein E, and then below flavivirus in the search. Go to the gene, not genome. Protein E flavivirus, and I'm going to tell you also how. Protein E, E for Eva, E for elephant. Protein in English is ABCDE. Yeah, E. Yeah, because you know, it's very difficult sometimes to pronounce this. In English, it is E. In Slovak, is A. Is E, and in French, for example, we say Japan. Spanish say Yapon. So J is here. In Portuguese they say kh, so kapon, and in Slovak we say yapon. But J is pronounced as J, Y, and K. How I can go to my screen is shared. Yeah, thank you very much. So look, for the downloading, I will go to NCBI. See here, NCBI nucleotide, for example, or gene, doesn't matter. So go to nucleotide. Then here you have search. On search, you have more option. Besides search, you had more options. Protein E, right? Protein E plus Flavi. Search. What happened? Okay, with gene I'm successful. So I have here polyprotein from Yokose virus, Usutu virus is also Uganda S virus, Zika virus. So all these are flaviviruses. Yeah. So you don't need to do it now, everything. I will just explain what to do. So I'm going to this download, okay, the same as you did for the primer. And one by one, after downloading, I'm going to put in my folder. Okay, so I will have 10, 15 different sequences. You got it? Don't do it now, it will take time. And then I will end up with sequences. A minute.
I'm beautiful and I want to sound only for being here today.
No.
One more thing on Subway.
And then I will have sequences one by one like this sequence. So you will have sequences one by one. Okay, and that sequences are like this as well. Just give me a second. So what you will have is you will have different sequences, one from Japanese encephalitis and so on. So each virus will have its own sequence. And what you have to do is you have to select all like this, Control A, select all, and you go to alignment and multiple align. So alignment and multiple align. And it will give you several options. There are several algorithms of the alignment. There is Genius algorithm of their own, but two universally accepted algorithms are muscle or Clustal W. Okay, so we traditionally use Clustal W, but nowadays I'm using Genius. So with this, it will align your sequence, all the sequence. And once it is aligned, for those who are interested in the phylogenetic analysis, there is simple trick to go. Here is tree, you will see, and just click on this tree. There are again inside several algorithms like Tamura, JHK, and Jukes algorithms. I am fond of Tamura Nai. And again, universally accepted algorithm is neighboring joining. And just I will put OK. And this will make very nice phylogenetic tree. Okay. So the same thing I can do with my sequences from what I get from the sequence file, FASTA file or sequence ABI file. The same thing I will show you. I can do that. So for example, if this is my sample, you will see its neighboring partners and from which country they are, and you can have you can also make different graphics of that and so on. So the same thing I can do with the files which I got from the sequencing, right? So these are sequence file. This sequence file looks like you have chromatogram, and the same chromatogram I can go for alignment, multiple alignment. Okay, you got the point. This okay, and then it will make tree, and that's it. So you will have here alignment tree. Okay, so for clicks and you are in phylogenetic analysis. So based on phylogeny is one thing, but for primer design we need homology and heterology, right? And phylogenetic analysis is based on that. So let's go back to homology and heterology. Sorry. I don't get.
You are going to get alignment for make two homologous sequence. How you select the other sequences from other country or homology on the basis of which parameter who criteria which criteria the other people they are doing work and make our sequence.
Yes, I will answer. Yeah, understand. So for that, first you have to blast. Okay, and you after blast, you are going to download one by one by one by one. Now, why I'm telling you or teaching you this software? It does it everything for you with one click. Okay, if you want, if you are interested, I can show you now. Okay, so for example, you have this sequence. So this is non-structural protein NS5 protein from, I don't know, let's see from where it is. Okay, so there is one trick for that. Here you see blast. In prime also you have blast, sure. So click the blast, and now there are several databases. Actually, I want to give you this lecture on Thursday, but let's see faster. So there are several databases. We have NR database. Details will be on the Thursday. There are several algorithms: blast N, blast P, whole blast, mega blast, and several blast, and so on. So what I will do is search, and then this window here has pop up. It will searching. Let's wait for the search. Better to wait for the search, otherwise Again, if I will disturb, then it will do it right from the beginning. Actually, there is also a way to do this. That means you have to copy the sequence, okay, and you have to put into the web server, NCBI. So actually, this is doing for you everything. Sorry. Battery. Okay, let's go ahead. I will keep it. There is some issue with because it's stuck to 48%, but I will come back, okay, to this point. So we'll go to the homology, but I'm sure I will come back. Don't worry for this. Thank you. Okay, let's go back to the homology and whatever we were trying for the primers. So in this slide, what you see here is different sequences, okay, of each virus. For example, this is a tick-borne encephalitis virus. This is Paso virus. Maybe these are the West Nile virus and so on. So the genome looks like this. Okay, so viral genome, this is from 1 to 11,901 base pair. So this is a genome of flaviviruses, more or less 100 base pair. It looks actually like this. Okay, but if I zoom in, it looks like this. I can do the alignment, okay, and make a phylogenetic, the same as I told you. And that alignment is also used to make the primer. Okay, now this software will allow you to identify the region which are homologous and heterologous. Okay, it's very important point. This software will allow you, and this will be denoted by color. So green color is homology and white color is heterology. Yellow color is less homology. Let's go for that. So see this line here. So this line represents the homology at that locus. Okay, I will go one by one and slowly. Okay, it's big picture now. So you see like this. So at this point, you have homology because there is amino acid the same and also GC sequence is the same. Okay. Now this is just one point. We will identify the region of interest, homologous region of interest. Okay. So a little bit, and I will go from the beginning. Okay. Homologous, but here sequences are not present. Slowly I will go. So you just follow this, okay? And tell me which region is good. I'm going slow. Okay, I will go a little bit faster now. Okay, here it started to have some homologous region, a little bit more. Again, heterologous region. Something good. See this again. Amazing here, right? Again bad. Let's go ahead. It will take time, but it's worth. This is good region again. Okay, and then I will go to the region which is really good. I had primers here somewhere. I'll just check where are my primers. Okay, so that region I will explain you in details. Yeah. So we are somewhere here. So in this whole genome, when I roll it from 1 to 9,100 base pairs, okay, I found here that this region is really good for me because this region has a maximum homology over here. Okay, and this region could be a region for my forward primer. Okay, and to make it generic, it should be span the heterologous region. So you should have homologous, heterologous, homologous, whether it will match. So we have found one homologous region. Let's go. Very nice. Homologous, start heterologous. And again, very high homologous. Yeah. So, for example, based on this red, I will be able to discriminate different species within the genera. I repeat, based on these regions, these red regions, I will be able to discriminate with sequencing different species within genera, right? And let's see how different they are. For example, this region is interesting because there is a lot of heterogeneity. So look here, GCAGG, GAAA. So based on these nucleotides, I will be able to tell which species and from where it came from. Okay. So idea is to able to amplify this region and then sequence and genotype. Okay. So that's why what I did is I selected this region as a forward region or forward primer. Now, unfortunately, I don't have much freedom to go for less dimers and whatever. So I have to tweak a little bit. Okay. So what I have done here is I have selected 20 base pairs, not 21, 27, because I'm restricted to the homologous region. Okay. And I saw that I will go here. Sorry. Yeah. So when I click here, it will give me the information, the same information what we saw in the morning. The rough TM is 49 only. Rough TM is 49. But what is rough? Because there are several sequences, is not a TM of one. So what you are asking, whether there is change in TM, this is answer, that this is a rough TM. GC content is again different. And I will not go into the dimers and cell dimer. I will just skip that. So 49 is the temperature. Let's go back to this reverse primer. Rough temperature is only 42. Now I have to decide, should I move this primer a little bit here or here to increase the temperature? I may increase, okay. But there is also one possibility how to increase the temperature. I will tell you later. So this region is interesting. And if I will design primer here, it will give me a product of, so let's see how big product. Yeah, product of 353 base pairs is enough for me. Okay, is enough to discriminate the species within the genera. So that I call this a generic primer, or also a pan-flavi. That means it will amplify pan-flavi DNA from different flaviviruses, and I will be able to discriminate them. Now, how to order this primer? Because there are several sequences. Here it comes the degenerated primers, what I told you in the morning. So you have in front of you computers, go to the Google and check IUPAC codes for nucleotide. And you will have table A, T, and so on. It is in Google in normal. You have? Now you will tell me if I want to order this primer, how I will order. Let's go. I will make it bigger. So you will see. Uh-oh. Okay. So now you will dictate me the sequence. How I will order the primer. A. Again A because you. Sorry. A. T. Sorry. T. G. G. C. And here is problem. Don't see this. This is M. See in the table. M because there is A or C, right? So while ordering the primer, I will put M. Next, A, T, G, A, C. Then there is A or T, that is W. Yeah, W you have. Then there is T, T, T. And then here is a big heterology is H. Okay. If there are 4 different nucleotides, then N. And you order the primer. Let's go back to this reverse primer. Reverse primer is nice. Homology, only here you have to put the degeneracy, or there is M, C, or A, and you order the prime. You got the point? Yeah. Now, because of that knowledge, what we saw today, if you add G or C, you increase the temperature. What I will see in the reverse primer, if it is only 42, the melting temperature, I will go here a little bit. I will add a C. My temperature will increase. I can add A and I can add T. The temperature will increase. Again, it will not be highly sensitive primer. It will give you some non-specific, but no problem. Okay, because for sequencing, what you are going to do is you are going to run the gel, you are cut the primer, cut the band, and then you can send. So you don't care about non-specific. Okay, but at least you will have some idea that which flaviviruses are circling around. You don't need to go for each and every virus and each and every multiplexing. So this is simple procedure. Okay, so sometimes it happens that it is a non-stable primer. Okay, because it degenerated primer, it could be it is unstable, or the TM is less. Fortunately, we saw here there is C, A, or T, but sometimes it happens that you have very less homology. For example, here in this space, yeah, here you see at this end you have heterology or heterogeneity. This end you have heterogeneity. You don't have much space to move around, right? So I cannot add more GC or interest of region over here. You cannot add much. So what should I do? So what I will do here is I will extract. Yeah. So this is extracted. For example, so this is your primer. Only one primer. I need only one, not degenerated. So here is something. So I don't need degenerated primer. Watch here. You have rough TM49, what we saw. Yeah. And I will increase the temperature. I cannot add any nucleotide in this region here because growth is from here to here. So I will put allow editing. Allow editing. And here I will put G, C, G, C, G, C. Look what happened here. 62. And this is called a GC clamp. Okay. So this GC clamp will increase the melting temperature automatically, and it will stabilize your primer. Okay. So let's add GC, 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 GC. $29. Just a minute. There are 40 bases exactly. So do you see this structure? Very weird structure of your primer. Okay. So do you know why I added this? Any idea? Sorry. Yeah, but increased TM is only if you add 4 or 5 are enough, but not 40. But this also is called as good GC clamp, and that GC clamp you can genotype your PCR product based on melting, the total melting, and that total melting, what will happen is this is your PCR product. Okay, this is GC. So PCR product will look like this. variable region, GC, 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 complementary CGC, GC, GCG. It is very hard to break because it's very high compact. So it will make a big hairpin like structure, right? And then in HRM, it will slowly unwind like this, but still it will be intact molecule. Yeah, and this phenomena is called as DGGE, or this is called as clamping of your PCR product for the genotyping. This is whole new experimental approach, but just to have an idea, if you put 40 GC, you can use it in different downstream application, and it is called as DGGE. So if you are interested in that in your laboratories or in when you will go back, you can also apply this kind of for genotyping. Is very fast. Okay, any question in this case? So we saw normal primer design, we saw degenerated primer. I would not go into the multiplex primer because it is simple as the normal, because you just combine different primers and you have multiplex. If you are lucky, you will have product. If you are not lucky, you have more non-specific than specific. So the simple primer design, degenerated primer design, and let's go for real time now. Okay. Yes. This GC clamp, it is in the GC clamp based high resolution melting analysis, or DGGE. You can also write and read later. So we can use for genotyping. Why I'm telling you, because you all came over here for the diagnostic approach. So the gold standard is sequencing always. That's why he got the Nobel Prize, Sanger. Then biotyping based on the protein. But let's go back to nucleotides. You have SSCP, single strand conformation polymorphism based on gel. You have HRM, high resolution melting analysis. You have DGGE, denaturating gel electrophoresis. You have ASA. You know what is ASA? Allele specific. To cover that actually. So you have ASA, and you have a lot of different. So this GC clamp is to stabilize and use in DGG. Okay. So as I have mentioned ASA, let's first check that and then go for real time. Okay. Now, actually, I wanted to cover ASA later, but as we have opened the alignment, I can cover ASA now. So let's go to alignment. And this is the best example for ASA, one of the best example. So I don't have to search again. So first approach is to make the degenerated primer. You all understand we want to have a mix of all primers in one tube. I want different approach. I want to have the same primer degenerated but specific for particular species. It is called as allele specific PCR or allele specific assay, ASA, allele specific primer. How it works? You keep the same primer. Your primer is working very well. You set up the PCR, everything going fine. You are able to amplify different genotypes and different species and so on. But in this case, I don't want to amplify. For example, if this is tick-borne encephalitis, I don't want to amplify tick-borne encephalitis. What I will do, the tick-borne encephalitis virus ends here with G. I will not incorporate, and I will extend this primer one nucleotide more, and I will order the primer, the whole plus A or T. A or T. So all the viruses, all the genotypes or all the species that ends with G will not amplify because G is not there. Okay. So that is called as ASA. You know why it is not amplifying? You know the reason behind that? Okay, I will explain. So in the morning we saw a DNA dependent DNA polymerase, yeah, and that is a key component in ASA. Okay. To be able to extend the DNA or polymerization, what is necessary? You have a nucleotides. Is DNA. Here, forward primer will annual, right? And if I have here, this is a T, and if this primer ends with G, will it extend? Will it extend? Will your PCR successful? No way. Why? Because this end is unattached. Okay. So DNA dependent DNA polymerase needs the 3-end complete. It will otherwise not grow. It will otherwise not fill in the nucleotide. Okay. Now, in case of ASA, what you have to do is, if you know that the tick-borne encephalitis at this position ends with T, you do not add A here, but you add all except A, right? So all genospecies which don't have T here will grow very nice. This will not grow. So this is a simple technique behind this. Okay, you got the point, Popasa? Okay, let's go to real time. If you have question, please ask. So theory will be simple for the real-time analysis. Yeah, I will finish the real-time and then it is very fast, this real-time. So real-time is theoretically the same procedure for normal PCR, normal primer design. Only you have to consider 2 or 3 things. First, your product should be as small as possible, around 100 to 120, not 50, but 120. So this is one point. The melting temperature should be more, around 65 to 70. Okay, so it should be more specific. In case of bacteria and in case of viruses. Okay, in case of parasites or eukaryotic cells, how many of you are interested in eukaryotic cells? Nobody. Yes, eukaryotic. Okay, so extremely important point is that what is difference between DNA of prokaryotic and eukaryotic? A fundamental difference. Sorry? Yeah, but it's mRNA or RNA is uracil. But at DNA level, what is the fundamental difference between prokaryotic or in bacteria and parasites? The parasite contains exons and introns, or eukaryotic contains exon and introns. Bacteria, they don't have introns. Okay, so this is a fundamental difference between eukaryotic and prokaryotic. Now, when you do the PCR, first you need to isolate the DNA, right? In quantitative PCR, for example, in case of eukaryotic, you want to amplify or you want to quantify the expression of gene, yes, or you quantify how many copies of that particular pathogen is or number. So in case of bacteria, unfortunately, what the hell is this? Unfortunately, you don't have any other choice, but you just go for forward and reverse 120 base pairs, for example, from between 120 base pairs, because there are no exon and introns. In case of eukaryotes, you have this structure: intron, exon, intron, exon, so on. Intron, exon, and so on. So if you want to quantify based on mRNA, what you need, the exon. You need exon and exon. Okay, you don't need introns. Okay, so if you want to quantify mRNA, your primer should be on one exon and reverse on the second exon. Just write down. If you want to discuss, I can discuss half an hour later, but I don't want to in between you and lunch. So just write down, yeah, just write down that for mRNA and expression level, you should have intron spanning primers. So one primer should be on one exon, another primer should be on another exon. Whole of the exon. Yeah, intron should be excluded. From the intron, one nucleotide is enough. It should not include any nucleotide from the intron. Yes. Yeah. Okay, so this is typical difference as far as quantitative PCR is concerned, real-time PCR is concerned. I recommend you to go for cybergreen based real-time PCR, not probe and fish based assays, because they are more complicated, more complex. Start with simple Eva green or cybergreen based assays. Okay. Details, I don't want to take much time. Reverse transcription reverse transcription PCR, those who are doing or want to do viruses and eukaryotes. And then I have one more for the ligation. If you want to have protein analysis, if you are not interested, then I will not cover that. Okay. That's it for now. I don't know. There are several other primers for hybridization, PCR, OEPCR, in situ, fish, fluorescent assay, and so on. So if you are interested in, I will cover fast in the afternoon. But if you are not interested, I will not cover that. Okay, it's up to you. Okay. Okay, fine. So we will end with primers. Any questions in that? No. So I will cover that, and then, okay, so for reverse transcriptase, what you have to have. So this is gene, okay, you have for reverse transcription, that means you have to reverse transcribe your RNA to cDNA, right? That is, you have to change your strand. And for that, there are 3 different ways. The mRNA looks like this and ends with and so on. This is called as poly A tail. Okay, that gives a stability to mRNA. And for reverse transcription, you need RNA dependent RNA polymerase or RNA dependent DNA polymerase or mix of both. Okay, so it depends on the chemistry, and you need a primer to do that, because all these RNA or DNA dependent, they have to have some primer to link. If this is joined, then I will grow, it says. So what I will do is I will order the primer TTTTTTT. Okay, so this called as DT primer or poly T primer, and that poly T primer will attach to a tail, and it will start to fill a complementary nucleotide to RNA. That is why it is called as RNA dependent polymerase. In case of PCR, it sees the DNA. That is why it is called as DNA dependent polymerase. Okay, so this is the first for the reverse transcription. No, only T. So you have to order maybe 15 times T. That's it. No, you are ordering the primer to convert your RNA to cDNA. Wait, and after that, you have now cDNA that you can amplify with PCR, because in PCR, yes, all human, parasite, or whatever. And then you order the primer which are specific to particular region, and that primer is the same as I taught you in the morning. Yeah, the same. Okay, you are also able to convert your RNA into cDNA using N. So 6N. This is called as random hexamer primer. Random hexamer. So you order, you send company 6 times N, and they will send you random hexamer primer. And that random hexamer primer is mix of all possible nucleotides. Okay. And this can, as this is random, if when this is random and this is cDNA, sorry, this is RNA, it can sit here, 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 anywhere. It's random. And once it sits, the RNA polymerase will start to fill in like this. Okay, so this is second type of primer that will convert from RNA to cDNA. Yeah, another possibility. You don't want to convert everything, you want to convert specific. It's very simple approach. Use the reverse primer in reverse transcription, and you convert only a specific sequence. Okay, so you have 3 primers to convert your RNA into cDNA. Once you make the cDNA, you can amplify as a DNA. Good question. You are hungry. Okay. So RNA dependent polymerase has some limitation. It will grow. Oh, it will grow, but not long. It will grow up to 1 kilobase, for example. But several genes, for example, factor H, toll-like receptor, and all of the some genes, they are very big. C4BP complement proteins, so they are big. Big means how big? 91,000 base pairs. Factor H. So not a single commercially available RNA-dependent polymerase can translate, right? So if your primers are here, you are not able to amplify because there is no CAD DNA. You got the point. In this case, you use the random hexamer. It will sit here, 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 everywhere. It will translate. Okay. But random hexamer will translate or reverse transcribe your all RNA copies, RNA species. Disadvantage. I want only mRNA, not other hRNA and pre-RNA and all RNA species. I don't want. I want only a mRNA because that will be translated into protein. So for that, all mRNA and only mRNA contains polyL tape. So if I use T, it will convert only mRNA. If I use random hexamer, it will convert everything. Yeah. So some advantage, disadvantage. My student, she mix both random hexamers and all. They are trying. Okay. So this is primers for reverse transcription PCR. Real time we cover, and normal PCR also we cover. Okay. I recommend you all to go through the software because that software we will need. Yeah. And your last question, I think you asked. I'm sorry, but I want to finish this and then have. So okay. So you remember this? We put for the blasting. Yeah. And we have here results. Okay. So I put for the alignment in the gene bank through this software some sequence. And that sequence is now, so this is my sequence. This is my sequence and this is the alignment. Okay. And this green shows me homology. Okay. So pairwise identity is 100%. And this fellow is West Nile virus, SK, Pi8, NS gene, and so on. So your question was how I will download the sequences to make the phylogeny, right? Which sequences I will, you asked, or who asked, you or you? I don't know. Okay. So what I will do is I will select this, or maybe I will select all, and I will put download. Yes, there are indicators. For example, I will check location. I will check from where they are. For example, I want to make some homology and I have in publication, I want to make the publication interested or interesting. That means, okay, this comes from this genotype is also in China, also in Japan. So I will select based on geography, for example. Okay. And that information you will have here in this. Okay. Or also you can go to one and information here one by one. You can check and you can put all the download. And after download, you will have all downloaded sequences. Make the phylogeny. Yes. Yeah, you can also have the query coverage. For example, here you have 99. So you can also take into consideration. It depends on your virus. If, for example, when it was COVID, it was maybe 98%, and then it started to have 100%. It depends on how new is virus, how remote is that pathogen. So, for example, parasites, they are not that homologous, but viruses, sometimes yes. It depends on what gene. For example, protein S, spike protein of COVID changing. So in that case, you can go at 99%. And if you will see that, oh, there is a lot big difference, not 99, but 96, then it is mutated. So it depends on what you want to show. But go between 95 to 100. 200. Actually, it is not my field now. I was doing in PhD this one. But yeah, 95 to 100 is optimum. Okay, that's it. If there are no questions for in this, yeah, yes. Yeah, I told you, I answered the question. I answered because he asked the same question. So both are good, all are good. If you want to amplify specific mRNA, then oligo DT. If you want to convert whole, then is random hexamer. Should we close now? Okay, thank you. Yes. Visit only.
So online participants, yes, now